It's a pleasure to be here. Thank you so much for the invitation. I would like to share with you guys just a little bit of a story we're trying to finish within the next month or so, if possible. That's an intervention using 2 different carbohydrates and how the microbiome in Brazilians, in this case, actually responded to those interventions. So, as everybody before me has presented you, we are composed by this collection of microorganisms, which make us actually a meta-organism, if you will. The idea being that we are living in symbiosis, that we are all utilizing the resources that we have available to us. So diet that we are thinking as human diet actually is feeding not just us, but all the bacteria that live within us, fungi, and the energy is actually propagating throughout all the trophic levels. If you break that, of course, then you enter in this state of what we would think as symbiosis, becoming dysbiosis, and leading to pathogenesis of some sort. Data, it's coming into the literature. The idea would be that if we know exactly how to deal with this interaction, we should be able to control it and use it to our advantage and eventually modify it as presented earlier by the speakers. And my personal interests actually came about a couple of years ago when the data presented this correlation between the so-called enterotypes and the consumption of carbohydrates, or a different enterotype and the consumption of fats and proteins. And to me, that seemed, striking, that there was the first time that we actually could see this relationship. And if that was so, then we should be able to start digging out who we should target with what kind of intervention to actually change. And within this framework, we started, this pilot study in São Paulo using a carbohydrate source that had been developed there. That's unripe banana flour. This is a very resistant starch-rich component. It's very abundant in Brazil. It's a waste product, for the industry. So we were trying to solve several problems when this was developed, and it is in the market today. You can see in stores, and people are using it with alleged benefits for their health. And there are some benefits. We have other data showing that there are some influence within the host's health. And our question was, what is this actually doing to the microbiome? How is this actually affecting the microbiome? So the clinical trial was designed to have a placebo-controlled and double-blind study, and we had 2 experimental arms. We had one arm with the resistant starch component and one with inulin as another carbohydrate that we know is not available to us and would reach the intestine. And this was a consumption that was supposed to be the least intrusive and the most realistic as possible. So the consumption was 3 times a week. It was delivered as an additive to soup. People would consume it at night, usually replacing a meal. And we conducted this for 6 weeks, and we assessed the microbiome of these people before and after the intervention. And the workflow is what people are familiar with when we're talking about microbiome studies. We actually take a fecal sample and measure the DNA out of this as a surrogate of measurement out of all the bacteria that are there. With this in hand, then we can do all sorts of nice analysis in the groups that are present, and what actually they're doing there. So the first question was who was actually there, since there was no data on Brazilian microbiome, as was presented here earlier. It's a group of people. It's a very large country, very diverse, with very different ethnic backgrounds and dietary components, if you go to the northeast or the south, and nobody had really studied what was going on in the microbiome of these people. So the first thing that we decided to see was, well, who is there, and can we find any patterns, and do they correlate to what is being published? And the first thing that we saw is that we have this very high prevalence of this Prevotella-rich enterotype within Brazilians. The number that's most often seen, or the most commonly seen for westernized populations in the US and Europe, is close to 10 to 13% of people being members of this cluster. And in Brazilians, we see here about 40% of the individuals that are actually members of this cluster. So what I'm presenting here is just a heat map, and in each row you have the genus that's being shown. The two very bright red genera, and you can see here, but yeah, you still can see. The one on the top that is on the left side of the screen in red is the one that actually are the Bacteroides, and the one in red on the bottom of the heat map is the Prevotella. And you can see this dichotomy as was presented prior in other groups that we actually have seen across the world. This was statistically significant. It was the same method that was actually used by the Meta-Heat Consortium when they published the enterotype paper several years ago, and it has been replicated in other groups and contested by others. Our question then, went on to see what happened to the intervention, and nothing happened as far as taxa goes. We didn't see any taxonomical changes. Groups of bacteria didn't change. But that doesn't mean that changes were not happening, because we did see effects within the host. Like I said, we had some publications on that before. And the question then becomes the function. We know, and this is the third time you're seeing this figure on the left, that the microbiota of people can be quite different, to their taxa that are present. They are all healthy people, but they have different proportions of bacteria if you just look at who they are there. The same samples that are being presented on the left on the panel are being presented on the right, and at that point we're seeing the metabolic capability of these communities. So clearly they're all the same type of community. We still have guts that perform the same basic functions. So it's expected that we have the same functions even though we have different bacterial groups inside of it. So we decided to do an analysis using a tool that's available to us. This was all 16S when we started to do this. We went on to 16S thinking that we would have results that were on taxa level. And what is available to us to do now is use bioinformatic tools. This tool converts the 16S approach into gene content. It predicts what would be the functional genomics of these samples. And then you can use several statistical methods to actually make comparisons, and this is very similar to what you would do, for example, with a microarray analysis. I'll present to you some of what we did using the CAG pathways, which actually map metabolic within their metabolic pathways, the genes within the metabolic pathways. The first thing that actually became apparent is that we only have an effect of these carbohydrates if you actually look at a stratified population. So if you take the cluster A of these people, the people that are actually Prevotella rich, then yes, we have a very strong effect of the resistant starch consumption. We see here almost 800 genes that actually are significantly altered by the consumption of the resistant starch, and we don't see any changes within the people that are, members of the other cluster. So if you're Bacteroides, you don't see any change consuming this resistant starch. And if you are a Prevotella member, then you actually have this change. We see a much more subtle change with the annealing group. Only 49 CAG orthologs actually changed as opposed to the almost 800. And please note that the numbers of individuals within each one of the clusters is different. We didn't assign the people to the arms knowing what they were. We found out what they were after they had been already assigned to groups and being studied. So this perhaps is something that we need to take into consideration when we're thinking of interventions and trying to determine something about the microbiome before you assign them to an arm of an intervention. This is a little bit more visually apparent here at this heat map, and you can see are the samples that were actually obtained from the individuals from cluster A only. They're presented on the large heat map, and you can see from time 1 to time 2 this very drastic shift from the dark colors on the bottom to the yellow ones on the left. And the same genes and the same order are presented for the cluster B people, and that's on the inset. And you can see that there is no pattern, no obvious pattern that we can actually see here. So among the things that we saw that actually compose this heat map, we have several short fatty acids being genes being activated and being upregulated, if you will. And at the same time, we saw some pathways that actually lead to the production of LPS being completely downregulated, entire pathways. And we're finishing the drill down to see what it actually means and which ones are the genes and what is happening here. Yeah, it's just an example of what is happening, and you can't really see, but this is a KAG pathway for the propionate metabolism, and you can see all the genes that actually lead to the production of propionyl-CoA being upregulated. The question then becomes what happens with the other carbohydrate source, and as I showed you, only 49 KAG orthologs were actually regulated for these people, and the most interesting part was that when you see what happens to them, they were actually all decreased upon consumption of inulin, whereas the same genes generally show the opposite pattern for the consumption when people consumed the unripe banana flour. And you can see just the categories being annotated on the side, and most of them are assigned to energy metabolism and carbohydrate metabolism. So we see this dichotomous behavior within people that are assigned to the same type of enterotype. They behave differently depending on what type of carbohydrate you gave them, even though those two carbohydrates are unavailable to the host and are arriving to the colon largely intact. So just to summarize everything that I said here quite fast, we see this modulation observed only for people that belong to the Prevotella cluster and not to the Bacteroides cluster. It's a very high prevalence of people within this cluster in Brazil, higher than was reported in the literature, but not the highest that we have seen in the world. If you take populations from Africa and other parts in Latin America, you find people actually are very, very much enriched in Prevotella amounts. We also see that resistant starch has a much more broad impact within the gut microbiome than the consumption of inulin, and that an apparently opposite effect actually is happening here for the same genes when they're actually shared there. So with that, I just would like to thank everybody that was directly involved in the project, the people at the University of São Paulo and also at the University of Pennsylvania, where we have collaborations for quite a while. My funding sources, so it's nice to thank the people with the money. Thank you.
Thank you very much, Dr. Hoffmann. This very interesting talk. Questions? For me, perhaps. Being not a microbiologist, I mean, we had this, as you mentioned, these Bacteroides and the other ones.
The Prevotella.
Yeah. Is that close to the firmicutes that Dr. Escobar had alluded to?
Those are Bacteroides, the Bacteroides, all within the same phylum, no firmicutes at all. Yeah, we didn't see any remarkable correlations with firmicutes on this intervention. We do have some, but they're more punctual. And
I see. Yep. Yeah, very interesting differential effects. What makes the difference then between the unripe banana flour and...
between the two carbohydrates?
Yeah. I mean, it's also inulin containing. Probably not as much.
Probably not as much. As far as we know, if it is there, it's very, very low. I don't know exactly why we have that. We have an idea of why this is happening only to the Prevotella group, but why we have such distinct patterns actually, and why it's so much more broad-reaching with an unavailable, just resistant starch, we just don't know. Perhaps it's just a matter of the enzymes that are actually being produced. Alpha amylases are more prevalent than the ones that are needed to break the beta bonds from inulin. So maybe something like that. We didn't see that signature though, that's speculation.
Okay, thanks a lot again.
Sure.